This is the 2022 second quarter online investor conference of Oneness Biotech Company, Limited. We are very pleased to be joined online by the distinguished guests and the investors in this virtual investor conference. First of all, I would like to introduce to you the management team: General Manager Qihui Cheng, Vice President of Medical Science Shizhen Chen, Vice President of the Antibody Development Nianyi Chen, Vice President of Small Molecule Development Nina Wang, Senior Manager of the Finance Department Amy Xia. Next, I would like to invite Qihui Cheng, the General Manager, to give a brief speech.
Dear shareholders, ladies and gentlemen, good afternoon. It is the second investor conference in 2022. The management team should report to shareholders on progress of the research and development of the new drugs of Oneness and the important aspects which shareholders pay attention to. We are hoping to let the shareholders know more about Oneness. In the second quarter of this year, the new drug R&D entered several key stages, including the data analysis of OB318 US Phase 2A clinical trial, China NDA review on ON101, the licensing of ON101 and the regulatory consultation of ON101, and the US IND consultation on SNS-812, the siRNA new drug for COVID-19. In the meantime, ON101 is under application for health insurance reimbursement in Taiwan, and the United States Medical Device Application Review are approaching the final stage. In addition, the NDA review in ASEAN countries and the EU MDR application are also being closely watched. Many opportunities are accompanied by risks, and the team continued to move forward by seizing opportunities, controlling risks, and taking on challenges. First of all, we would like to share with shareholders that Oneness has been ranked among the top 5% of the Taipei Exchange listed companies by the 8th Corporate Governance top 10% of listed companies with a market capital of 10 billion Taiwan dollars or more in the non-finance and non-electronics industry for its corporate governance performance. This is an acknowledgement of Oneness' dedication to safeguarding shareholders' rights and interests, strengthening the structure and operation of the board of directors, improving information transparency and implementing corporate social responsibility. This is also the most important corporate responsibility of an R&D-driven biotech company. Secondly, OB318, an important pipeline of Oneness, was impacted by the epidemic on the clinical trial where targeted patients were affected and primary endpoint was missed. However, after a complete analysis after data unblinding, OB318 showed comparable efficacy to the global competitor in the target population based on scientific rationale, which is consistent with the results of 2 exploratory trials completed in Taiwan. The CSR of this Phase 2A study has been completed and delivered to the partner. The $33.6 million upfront payment will be recognized. Subsequent clinical trials will be discussed with the regulatory authority and the partner. The new drug development process is never a smooth path. The 2A study was signed to explore all the possibilities or even risks in order to provide references for future Phase 2B and 3 trials. Therefore, regardless of the external opinion, Oneness is ready and has sufficient capability and cash to accelerate the development of OB318, and is confident that it will succeed in subsequent trials. OB318 is positioned as a new drug for atopic dermatitis that rivals Dupixent, which hit a $6 billion sales last year. For the NDA of ON101 in China, Oneness has submitted all the dossier and supportive information. There was no DFU new drug applied for NDA in the past. It is necessary to continue the communication with the health authority in technical perspectives of DFU. We are hoping that the review keeps on the basis regulations and science and issues will be resolved soon. We also work with a CRO to initiate the Big Bay Area progress. This area, together with Macau, will be under a joint commercialization plan. We want to be prepared for all possibilities. As for the out-licensing of ON101, we have been positive and confident. Collaboration plans have been under intensive discussions along with regulatory consultation for NDA. We will continue to move forward and achieve our goal. SNS-812, another new drug to Oneness pipeline, received guidance from the FDA on IND planning this month and also received the attention from the state key laboratory in China. The in vivo studies have confirmed its mechanism of action in treating and preventing a broad range of COVID-19 virus, and have the potential to become an internationally competitive new coronavirus drug. On the basis of three major bases of science, integrity and transparency, the quarterly Legal and Theory will provide shareholders with complete and correct operating reports as much as possible. However, the accidents in the research and development of new drugs and the requirements of the regulatory units often occur in the unpredictable. We are good at mastering the risks to solve the problem, but it takes time. Please be patient and support Oneness. Continue to move towards a harvest and success. Thank you. This is the disclaimer of this legal lecture. Please refer to it.
Next, the research and development progress will be presented by Dr. Shai-Jen Chen, Vice President of Medical Science.
Thank you. I will update the R&D progress. The development progress of Oneness pipeline includes OB318, an antibody drug for anti-SEM-X, which is currently under phase 2 development in atopic dermatitis and allergic asthma. The second is OB704A, an anti-IL-6 antibody drug undergoing a phase 2 trial and the development of a second indication. The third is ON101, which is a macrophage-regulating drug. The presentation will include the update on Phase 3 trial in the United States, the NDA in China, the medical device application in the United States and Europe, the NDA in Asia, the publication progress, and the cooperation from academic, medical, and industrial perspectives. The fourth is OB318, an anti-cancer drug that is currently undergoing a Phase 1 clinical trial. The final one is SNS-812, a small interfering nucleic acid drug that is currently in preclinical development. For the progress of the antibodies, I will hand over to Dr. Ningyi Chen, our Vice President of Antibody Drug Development, to give us an update.
Thank you. Now, we will report to you the progress of OB318 anti-SEM-X new drug in moderate to severe atopic dermatitis Phase 2A clinical trial in the United States. We have completed the enrollment in July 2021, with a total of 99 atopic dermatitis patients. All subjects completed their follow-up visits and blood sample collection in January of this year. In March of this year, all biochemical tests were analyzed. This May, the statistical analysis after data unblinding were also smoothly completed. In June, this month, we have completed the clinical study report and delivered to the international partner for reference. Therefore, the current upfront payment of $33.6 million will be recognized. Next, I will report the results and analysis of the Phase 2A clinical trial in the US on moderate to severe atopic dermatitis. In this clinical trial, based on the analysis of biochemical values after the completion of the trial, we found that a total of 2/3 of the subjects were below the anticipated biochemical value of the target population. And we referred to a series of literature and found that in terms of biomarker value of moderate to severe atopic dermatitis, these 2/3 are different from with the typical profile. In particular, two key biochemical indicators including TARC, which is thymus and activation-regulated chemokine, and IgE, the immunoglobulin E. Both biochemical indicators were usually very high in atopic dermatitis patients based on the understanding of the disease. In the following chart, we have shown two clinical trials in Taiwan and the competitive drug Dupixent, under the same similar clinical trial design and the baseline IgE and TARC values of patients. The table with blue background is OB318 with baseline value for patients in the US 2A study compared with other trials. As you can see, in the lower right corner, Dupixent in the similar trial design, but the level of IgE is about 2,451 to 10754 IU per ml. The part at the TARC level is in the range from 1953 to 6147. In this range, we noted that the two clinical trials of OB318 in Taiwan fell in the range of similar IgE value and the TARC value to Dupixent. This shows that as long as we received patients who were under typical atopic dermatitis profile, the value is within this range. In contrast, our Phase 2A clinical trial in the United States may have been affected by the COVID-19 pandemic, resulting in the change of patient visits and profile. We noted that the value of IgE has deviated from the average, and this phenomenon is also seen in the TARC part. Therefore, these low biochemical values represent that these patients are very atypical. With the above subjects who did not meet the blood biochemical characteristics of the target population, we were able to reach the efficacy endpoints with statistical significance. With these patients added, there is no way to evaluate FB825 efficacy among patients' non-target profile. So we look further, through the literatures and discussion, to see which biochemical indicators can clearly help us differentiate these patients. We found that 1/3 of the patients with a relatively higher TARC value. The review articles or even in Japan's health system agreed on the use of TARC value to justify the patient profiles. Therefore, we use TARC to distinguish between 1/3 of the patients whose blood biochemical value profile was more in line with the target patient profile. For this 1/3 of the population, we can see two important key endpoints, including EASI-75, also known as the EASI score improvement by 75% in atopic dermatitis. This is the main efficacy endpoint to be evaluated generally in the Phase 3 clinical trials in atopic dermatitis. In this indicator of EASI-75, we will find that it is similar to the results of the Phase 3 clinical trial of competing blockbuster drug and to those of the exploratory trials in Taiwan. In the following figure you can see the green bar, that is, FB825 in treatment of the 1/3 patients with target profile reach is 53.8% in EASI-75. For the competitive blockbuster drug, its two Phase 3 trials showed 44% versus 51%, respectively. As long as the right target population is included, the treatment effect of FB825 can be compared to that of competing blockbuster drugs. Another key endpoint called IGA is the key to assess whether the IGA score of a patient can be reduced to 0 or 1. Based on this efficacy analysis, the results of the Phase 2A clinical trial of FB825 in the United States showed that IGA were reduced to 0 or 1 point in 36% of patients, compared with 38% and 36% respectively in two Phase 3 trials of competing blockbuster drugs. Therefore, combined evaluation with the main efficacy endpoints of these two Phase 3 clinical trials, FB825 can achieve similar clinical results to those of competing blockbuster drugs in the target group. In addition, we reviewed two clinical trials performed in Taiwan. With patients of atopic dermatitis under similar patient profiles, we will find that these two exploratory trials include the mean change from baseline in the EASI score and the proportion of patients reached EASI-75. The results in both clinical trials are very similar, which means that FB825 can show a consistent and stable effect. Reviewing the results of the clinical trials of FB825 in moderate to severe atopic dermatitis, we have the following summary. The Phase 2A clinical trial in the United States was affected by the COVID-19 pandemic, so it accommodated 2/3 of patients whose blood biochemical values did not meet the target population. Due to the addition of these patients, the main efficacy indicators specified in the plan have not reached statistical significance, and this part has been announced in accordance with the regulations. However, after clarifying the biochemical values of the blood sample, in 1/3 of our target group, the efficacy of FB825 compared to our control group is clear, and after sensitivity analysis on the key efficacy endpoints, including EASI-75 and IGA 0 or 1 score results, which are generally evaluated in Phase 3 clinical trial as primary endpoints, FB825 demonstrated similar data with the competing blockbuster drugs. In addition, the FB825 has another advantage, that is, its frequency of administration, 1 injection per month, and even our clinical trial in Taiwan can be extended to 1 injection every 3 months. The frequency of administration is an advantage. In addition, the safety of FB825 also has a very obvious advantage over competing blockbuster drugs. In our clinical trials, the drug's safety is well supported. In 1/3 of the patients, the target group, and the exploratory trials completed in Taiwan, these efficacy indicators showed a consistent trend. In these trials, we once again verified the mechanism of action of FB825, especially this mechanism of action is very novel. It is also the only novel drug in the world that targets the IgE expressing B cells. We have repeatedly verified the mechanism of action of this drug in these clinical trials. So in the overall evaluation on FB825 for the target patients, it has achieved our expected therapeutic effect. We will consult with the US FDA for follow-up trials to continue the development of FB825 in moderate to severe atopic dermatitis. Another important milestone in the development of the drug in FB825 is the development of subcutaneous dosage forms. Since clinical trials are currently conducted intravenously, we have further updated the formulation to subcutaneous dosage forms. Oneness has completed the development of the subcutaneous injection dosage form, and this dosage form development has also been acknowledged by US FDA through the Type C meeting. IV can be converted into SC dosage forms. The formulation of the dosage form has also been patented worldwide, giving a global patent protection on FB825. The advantages of subcutaneous injection dosage forms are as follows. The subcutaneous injection dosage form, due to its convenience, can greatly increase the acceptance of patients. In addition, during intravenous injection, the patient must administer the drug in the injection chamber for more than an hour, while the subcutaneous injection dosage form only takes the patients a few seconds to complete injection. Even in the future, when safety is further verified, patients can take the injection home and self-administer. This convenience can greatly increase the acceptance by patients, and in the subsequent clinical trial, it can also help reduce the barriers to clinical trial and accelerate the recruitment of clinical trials. For the subsequent plan of FB825 in moderate to severe atopic dermatitis, Oneness will proceed to consult the US FDA for a correct path and a more correct design. At the same time, we will prepare the production of subcutaneous dosage forms of drugs, and prepare subcutaneous injection dosage forms and intravenous dosage forms for the clinical PK study. And after confirming the PK data, Phase 2B and Phase 3 will be planned. The above is the follow-up plan for FB825 in moderate to severe atopic dermatitis. Another indication targeted by FB825 is moderate to severe allergic asthma under a Phase 2A clinical trial in Taiwan. The trial has been initiated at 4 medical centers in Taiwan. In order to speed up the enrollment, we have also prepared 3 other medical centers with IRB approval obtained. These centers will be able to start enrollment once the approval from TFDA is received. Since allergic asthma is a respiratory disease, the current pandemic is still at its peak, and medical centers have generally set up special COVID-19 wards in response to the pandemic, so the original medical capacity has been reduced and impacted the trial enrollment. Therefore, under the influence of the pandemic, we watch closely on recruiting the right patients with quality data as the most important goal. We will also closely work with the hospital to improve the recruitment strategy and patient tracking. The trial is scheduled to be completed in H1 in 2023, and the entire trial period is 9 months, so the goal is to complete the CSR in 2024. This is the arrangement for our clinical trial of moderate to severe allergic asthma. The next antibody new drug, FB704A, is an anti-IL-6 drug. We have currently initiated 4 medical centers in our severe asthma Phase 2A clinical trial, and we are also adding another medical center for patient recruitment. Once this center is approved by TFDA, the patient recruitment can start. The severe asthma is also a disease of the upper respiratory tract, so the trial progress has been impacted by the pandemic and patient enrollment was also be affected. During this period, in order to ensure that the correct patients are recruited and the mechanism of action is better understood, we also launched an exploratory study on the biomarkers by collecting the patient's samples. IRB review was completed for this part, and now we can start sputum collection and blood sample collection from patients to explore the responders to the drug as soon as the COVID-19 outbreak has calmed down a little. This exploratory trial is scheduled to complete the analysis of specimens in the second half of 2022. The Phase 2A clinical trials is still planned to be completed in the first half of 2023, and this trial period is 4 months, so the goal is to complete the trial in the second half of 2023. In terms of the second indication of FB704A, we have targeted the improvement of chronic kidney disease-induced cardiovascular disease. In this indication, we are actively conducting animal studies to evaluate that FB704A is effective in suppressing key indicators. In our animal studies, we have seen that FB704A can inhibit the key indicators of chronic kidney disease, including hepatoglobulin and neutrophil reduction, and has a very good dose relationship. So in terms of mechanism of action, we are currently getting preliminary certification. In another longer-term tox study, FB704A also completed a 26-week long-term monkey toxicology study, in which no other new safety concerns occurred and was in line with our expectations. After the completion of this experiment, it can fully support clinical trials of various lengths, so that allows FB704A with more duration for exploration. Oneness will perform a comprehensive assessment based on the data from in vivo and in vitro studies and the status of competing drugs on the market. We are scheduled to complete the evaluation in third quarter of 2022 to decide whether to include the improvement of chronic kidney disease-induced cardiovascular disease as the second indication. The above is the report of our antibody new drug. I will hand over to Vice President Chen to continue to report on other drugs.
Thank you, Dr. Chen. Next, I will report on the progress of the ON101 Phase 3 clinical trial in the United States. We have completed the first stage of the enrollment evaluation of 15 clinical centers. In order to have a smoother enrollment, we continue to perform the optimization of the clinical center. The CRO company also screens the new centers at the same time. More importantly, we work with Professor David Armstrong, an international KOL on diabetic foot. Professor David Armstrong is a renowned leader on diabetic foot research and ever published an article on DFU on New England Journal of Medicine. With the cooperation with Professor Armstrong, I believe that we can make our study in the United States progress as planned. The collaboration with Professor Armstrong included evaluation and recommendation of the clinical centers and the connection between American physicians and the International Medical Association. The most important thing in the U.S. Phase 3 trial of ON101 is to obtain the NDA in the United States, so we continue to discuss with the U.S. FDA on subsequent NDA-related matters. The relevant plans for proceeding to studies or request of waivers will be carried out in accordance with the requirements of the FDA. The overall goal of NDA in the U.S. by 2025 has not changed. In China, for the progress of the NDA, the comprehensive evaluation and technical review of Fespixin has been completed. On April 24th of this year, we received a supplementary notice for the NDA, and in response to the content of the supplementary opinion, Oneness has submitted a consultation reply for the supplementary notice on May 22nd this year, and has sent 3 batches of samples to Beijing, waiting for the Beijing Central Inspection Institute to accept for inspection. Supplements for other technical information will be completed within 80 working days. In terms of the global market entry of Fespixin, everyone knows that Taiwan's pharmaceutical market only accounts for 0.4% of global business, so accelerating global market entry is a very important market expansion of Fespixin. First, in the progress of market access in Asia, we plan 3 stages. The first wave includes Singapore, Malaysia, and Thailand regulatory submission. The Singapore's HSA has accepted the NDA application in the last December. The abbreviated review period is 180 working days. In February this year, Malaysia's NPRA also accepted our NDA application. The entire review period is 245 working days, and samples are sent for inspection in June this year. In June this year, we also received a product classification notice from Thailand, and we will apply for NDA based on the classification. We have received supplementary requests neither from Singapore nor Malaysia so far. The second wave includes India, Vietnam, the Philippines, and Indonesia. An application for a waiver for clinical trials was submitted to India in December last year for a review period of 6 months, and after the grant of waiver, we will be able to proceed with NDA submission. The waiver application is going on, and we are currently awaiting notice of review. For Vietnam, we continue to communicate with the Vietnamese DAV, and the documents are scheduled to be submitted in the second half of this year. For the Philippines, in April, the Philippines FDA has accepted our NDA application, and the review period is 254 days. For Indonesia, we confirmed with the local laboratory on sample requirements, and NDA submission is scheduled to be made in the second half of the year. The third wave will start in the second half of this year. An application for clinical trial waiver will be submitted in South Korea, and after the grant of waiver, an NDA submission can be made accordingly. The number of administrative review days in the above countries is provided based on the information specified by the regulatory authorities, but the actual number of review days will be subject to the requirement by the local health authorities. In terms of the progress of wound management medical device in the United States and Europe, US 510(k) application has been completed with supplementation submission and the reply from the FDA has not been received. The review time is generally about 90 calendar days, with replies within 30 calendar days after the completion of supplementation. However, this period is not a fixed timeline, and in the first half of this year, for example, the target review timeline of the United States 510(k) is 108 calendar days, so our US 510(k) case is still within the review period of this target. The MDR submission in EU will be made under the indication for scar care. In terms of quality systems, we have obtained ISO 13485 Quality Management System certificate. For MDR, the EU MDR registration will be applied in the second half of this year. OB318 is our new anti-cancer drug, undergoing the Phase 1 trial. The low-dose cohort has been completed, the second dose cohort has also been completed, and the Safety Assessment Committee has confirmed it is safe to proceed to the next cohort. At present, the subjects are being enrolled into the third dose cohort. Due to the impact of the COVID-19 pandemic, the attending physicians of each hospital must support the COVID-19 infection special ward, so the entire enrollment timeline has been impacted by 1 quarter, and the third dose cohort will be completed in the third quarter this year. Speaking of the COVID-19 pandemic, everyone knows that this has caused a great impact on Taiwan in recent months, so our nucleic acid new drug has been highly expected. According to the retrieved database on June 10th, the drug coverage rate of SNS-812 to the new Crown variant remained at 99.8%. For the new variant of Omicron's BA2 or even BA4 and BA5, SNS-812 still does not have any mutations in the position of action, which is mainly different from the vaccine. The vaccine acts on S protein, and SNS-812 acts on the gene location of the virus, so it can maintain such a good effect. At present, the drug synthesis, purity, and data of cGMP production have been completed, and subsequent filling have been initiated, and the GLP toxicological studies has been completed, and no obvious drug-related toxic reactions have been seen. In the preparation for IND application data, the pre-IND communication with US FDA continued. The US IND is scheduled to be submitted in the second half of this year. After the IND approved by the United States, an expedited review will be applied in Taiwan. In China, we have been in talks with viral research institutes to develop preventive drugs in line with local zero-COVID policies. This is the entire SNS-812 nucleic acid IND planned for COVID-19. This is an aerosol-type therapeutic dosage form, and on September 10, 2021, the first application for the pre-IND meeting was filed with FDA in the United States. On November 21, 2021, we also received a response letter from the US FDA, giving us a number of experimental exemptions and recommendations with an agreement on exemption of using primates in the non-clinical studies. This allows us only to use the Syrian hamster as a representative species for toxicology studies, which can accelerate the drug development. From December 2021 to January of this year, we also completed the signing of CMO and CRO. On April 30th of this year, the hamster's toxicology and dose-finding studies have also been completed. On May 3rd, we held a second pre-IND meeting with the US FDA, and received a reply on June 15 that many studies can be exempted, agreeing the nasal spray dosage form can be supported with aerosol-type toxicological data. This dosage form is more convenient for the application for clinical trials. Overall, we expect to be able to apply for an IND in the second half of this year. For the nucleic acid drug, our follow-up research and development progress includes the IND application for the aerosol type and nasal spray form, which is scheduled for the second half of this year. The aerosol dosage form is targeted to be completed as quickly as possible for the Phase 1 study in safety and preliminary efficacy evaluation in the second half of this year. Following the zero-COVID policy, the nasal spray form will be developed under collaboration with the National Key Laboratory and plans to obtain approval to proceed with clinical trials in the second half of the year by referring to the IND greenlight by the US FDA and the safety data obtained in the Phase 1 study. This will be first-in-human use of SNS-812 as an innovative drug against COVID-19, and everyone has high expectations, but the overall development schedule still depends on the review timeline of the regulatory authority. For the market sales of Fespixin in Taiwan, we provide the following updates. By the second quarter of this year, we have completed the listing application at all 21 medical centers. All physicians can make a prescription for the in-hospital pharmacy collection so that patients can receive the cream conveniently. In terms of regional hospitals, there are a total of 75 regional hospitals for wound care, and we have completed 55 listing applications, so the channel coverage rate has reached 73.3%. There are a total of 53 area hospitals provides wound care, and we have completed 39 listing applications covering 73.6% of the area hospitals. We also have covered 478 clinics and pharmacies as of today. Affected by the epidemic as a whole, many hospitals are supporting the fight against the pandemic, so many drug committees and application schedule are also slightly delayed. We will keep on our hard work and accelerate collection of drug use experience to support sales expansion so that more doctors will prescribe Fespixin to more patients and increase the market share. At present, Fespixin is still a self-paid drug, so entering health insurance is a great benefit for diabetic patients in general, especially for low-income diabetics. The reimbursement program is also highly anticipated by us. In April, we obtained CDE's Health Technology Assessment Report, called HTA Report, which is an important one for applying for health insurance reimbursement pricing. The review of the report is that Fespixin is a breakthrough innovative drug, which has the pharmacoeconomical advantage compared to the existing standard treatment. With the HTA report, the next stage is the expert meeting organized by the NHI. We will reply as soon as we receive expert meeting feedback. After these, the next is the pricing resolution meeting. If the pricing is confirmed during this meeting, Fespixin will be reimbursed by the National Health Insurance. There has been no drug treatment for DFU reimbursed by health insurance, and once Fespixin is included in the health insurance, it is expected that the burden of patients will be reduced and the majority of diabetic foot patients will be benefited. We also vigorously promote the cooperation plan between the medical centers, the universities, and the Research Institute of Fespixin, and encourage their participation in the academic and clinical research of Fespixin for value expansion. Current collaborative programs include venous leg ulcers, scars, biofilms for wounds, pressure sores, and the mechanism and treatment of DFU. Below is a summary of the programs we work with each medical center. In terms of venous leg ulcers, the safety and potential efficacy of the treatment by Fespixin cream in VLU patients are evaluated, and the current trial is ongoing. The SCAR program includes two parts. The first is the use of Fespixin on patients after surgical removal of thyroidectomy in the neck for the short-term and mid-term effects in improving scar. This trial is also ongoing. The second part is to use translational medical methods to explore whether Fespixin cream and other active ingredients can inhibit the formation of scars, especially the formation of hypertrophic scars, the microenvironment of scars, and the mechanism of regulation of chemotactic epithelial cells. This study is currently ongoing, and any positive results will be considered for a publication in the future. For biofilm in chronic infected ulcer, the animal model of biofilm is established on the diabetic animals, and the efficacy of Fespixin cream on biofilm is also being evaluated in this study. In terms of pressure sores, an open-label trial has been initiated to explore whether Fespixin is effective and safe for patients with sacral pressure sores. This trial is also being successfully started. As for the DFU, we hope to further explore Fespixin on patients under dialysis. This study evaluates the safety and efficacy of Fespixin in the treatment of DFU patients under dialysis. The included ulcers are classified in Texas Grade 1A and 2A. This trial is also being successfully implemented. On the other hand, we also hope that Fespixin can be used on more severe conditions, such as Texas grade 3A, 3B. We want to know if it can be used for downgrading ulcers. We explore whether the effect of Fespixin for such patients can be as good as on other grades. This trial is currently in the hospital's IRB revision review. Another new indication we hope to explore is radiation dermatitis. Experience in post-market exploratory use, according to physician feedback, has been shown to have good protection and reduce the severity of dermatitis. Therefore, in the second season, an animal model of this disease was also established to explore the efficacy and mechanism of action of Fespixin. From the chart on the left, you can see that the blue line stands for the mice which has not been given any drugs after radiation therapy. You can see that its radiation dermatitis occurred relatively quickly, and the severity was relatively high, and the recovery rate was slow. The red curve stands for a steroid that may generally be used to treat radiation skin cancer, although the steroid drug can reduce the skin severity, but the time of occurrence and the recovery were not as good as Fespixin. That is, the green curve at the bottom, the time of occurrence is delayed, the severity is improved, and the recovery was accelerated. We are very excited about this animal experiment, and in the third quarter, we will consult with CDE about the procedures related to the registration of new indications, and we will also hold expert meetings to determine the trial design of the entire study. If confirmed by experts, additional clinical trials will be initiated. Oneness has also been actively participating at various international and domestic conferences, including the key conferences, and continues to connect with the medical community and the international market. For example, in June of this year, we made a poster publication during the annual meeting of the American Diabetes Association. In September this year, we will participate in the presentation at the conference of the Taiwan Society of Burns and Wound Care, and at the end of December, we will participate in the symposium of the Taiwan Plastic Surgery Medical Association for a presentation. For the publication planning, in addition to JAMA, the mechanism of action via in vitro and in vivo studies on ON101 was also published by JID Innovations in June this year. ON101 is currently conducting a number of investigator-initiated clinical trials, and we will decide how the studies will be published based on the results after the trial is completed. ON101 has also been performed with cost-effective analysis and also conducted post-hoc analysis for its Phase 3 clinical trial. These data are scheduled to be submitted to international journals in the second half of this year. At the same time, we are also compiling FB825 Taiwan exploratory study data and expecting to submit them to international journal in the second half of this year. In terms of international cooperation and licensing discussion of ON101, we adopt regional licensing strategy, and the ongoing negotiations will be aligned with the progress of regional market access based on most favorable negotiation niche. For the major markets, especially Europe, the United States and China, there are currently a number of pharmaceutical companies with well-presented sales capability to propose terms for cooperation. The details of the negotiation can't be made publicly. Fespixin is close to the market. The timeline of the market approval is the key topic of the negotiation. We will continue to communicate with the regulatory authorities to accelerate the access to market. As mentioned by our general manager, the performance of Oneness in ESG has been ranked among the top 5% of listed companies in the corporate governance and the top 10% among the non-financial electronics listed companies with a market value of more than 10 billion Taiwan dollars. With the integrated development of sustainable environment as the main focus of environmental protection policy, Fespixin has also ISO 14040, ISO 14044 certified for life cycle assessment and ISO 14067 certified for product carbon footprint in 2022. In June this year, the first third-party verified ESG report has been published, disclosing the operation with credible content and data. This is our continuous enhancement on the implementation of ESG. Finally, we still have to remind all investors that Oneness develops global innovative drugs. Innovation is also accompanied by the high risk. Any research and development or trial may not progress as expected. Oneness has implemented a variety of risk control and management on the plan, but investors still need to be aware of the risk. It will take a considerable time to reach the stage where the new drug is successfully launched in the major markets with the establishment of the sales channel, inclusion in the reimbursement scheme, and acknowledgement by the physicians on its efficacy. Finally, we believe that Oneness has talent, capital, technology, strong momentum for innovation, a research and development projects for new drugs, and more. Determined to keep moving forward. However, it still needs to reach a turning point for profitability. The turning point include the milestones of the licensed drugs, the market entry of new drugs into major markets, and the continual licensing of drugs. Thank you.
Okay. Thanks Dr. Shaijian Chen for the presentation. Thank you very much for participating in this investor conference. We will call it a day here. Questions which have not been answered during the online meeting will be provided with a reply accordingly via the investors' Q&A before the start of the stock trading tomorrow. The next conference will be scheduled to be in late September. We wish you all a successful investment. Thank you.